cordgrass in California. Do you see this picture? Please tell me that you see it.
Yes, I see the
Yes.
picture.
Okay, so to give you an idea of the size difference between the East Coast cordgrass and the California cordgrass, this is Katie Zaremba. She is 5'6". So the California cordgrass comes to her knees and the smooth cordgrass comes to her waist. So I said all that. Now these pictures, and you can see the pictures, tell me if you can't, that shows the accretion of sediment that is due to Spartina alterniflora. Sediment moves through the tidal waters and is trapped in the plant, and then gradually the plant grows into those sediments and results in this elevation. This is a single plant in Willapa Bay, Washington, the original plant, and you can see how much higher it is than the mudflats. These plants coalesce and form these vast meadows. Spartina reproduces by both seed and vegetative growth. It is wind pollinated, mainly outcrossing in the native ranges due to early acting inbreeding depression that kills embryos. The seeds float and spread on the tide. There is no seed bank. In vegetative growth, individual seedlings grow into these circular clones equally in all directions in this unstructured mud. So what looks like these bacterial colonies are actually individual clones of Spartina. Hybrids were first identified in San Francisco Bay by Don Strong and his student Kurt Daler in '97. And then I came on board and did the first Baywide survey of using rapids. Now I'm going to step sideways here to talk briefly about the genetics of hybrids. These are using microsatellite markers. What we found is that there's been no speciation event. The hybrids have the same chromosome number as the parental species, that is about 60. We found through chloroplast DNA that both species were parents, seed parents to hybrids. The hybrids bred amongst themselves and backcrossed with the parental species, a process known as introgressive hybridization, and it resulted in a genetically diverse hybrid swarm. So each one of these bars in a structure graph, I should say, this genetics technique takes a 200 by 30 matrix. So all those elements are reduced to 4 colors, which is very cool. So each one of these bars is a separate individual, and you can see that some of them are made up of many different colors. These individuals are grouped together into general genetic similarity. What we found, so let's look at these colors a minute. The native species is blue. Spartina alterniflora from the introduced population in the mid-Atlantic is red. And in our work, we found another Spartina alterniflora introduction at Alameda Island that we don't know where it came from. We do know that it was the Army Corps that planted it there, but we have no record on where they got those seeds. What this shows is this is the alterniflora mother, and that all of the plants on Alameda Island, both established plants in the meadow, out on the mudflat, and these are 2 successive years of seedlings, all are related to this alterniflora founder. In Hayward, similarly, we see these mixtures of colors that are reflected in the seedlings in 2003 and 2004. So this is known as genetic structure, where individuals that are close together spatially can also be close together genetically. At Roberts Landing, we saw something very interesting. We have a meadow there, and then these isolated plants that grow out on the mudflat. And what we found in both 2003 and really by 2004, and we did the parentage analysis to confirm this, is that a single isolated mudflat plant, denoted in green, was the mother and the father by self-pollination of these green individuals out on the mudflat. And in fact, 2004, it contributed 30% of the seedlings that we found. So seedlings are locally produced, sometimes by selfing and dispersed. Okay, to step back into the field with it, this is a Foliosa marsh in China Camp, and it's about as tall as this gray blue heron. This was a hybrid invaded marsh in San Bruno, north of the international airport, and it's 7 feet tall. You can see, using Katy as our standard, it is far taller than Katy is. One biologist called this the day of the Triffids, this invasion, if you've ever seen that movie. Of course, these plants don't eat people. So the way we tracked the spread of these hybrids was through walking through the marsh, kayaking through the marsh, and bringing samples back from set locations and subjecting them to, we were able to readily distinguish hybrid plants that had the markers for alterniflora and the markers for foliosa in a single individual. Using those genetic markers and the maps, we found there was a spread of hybrids near the original introduction sites and across the bay, not much in the South Bay, not much in the North Bay. But the marshes where we found hybrids, they dominated those marshes. And then 5 years later, we found that the hybrids in green had spread widely down into the South Bay, all up the east and west sides of the bay, and into the North Bay. So again, stepping to the other side, I want to introduce the concept of exponential growth, which we should all be familiar with now, having seen this happen over the past 2 years with COVID-19. Small numbers increased dramatically, and actually in COVID, they fell dramatically as well. They did not do so in the Spartina. So how does this play out? We look at Willapa Bay, Washington, where there was only Spartina alterniflora introduced in 1900. So this is called a log linear plot. The year is linear. And the hectares of Spartina, the area of Spartina, is a logarithmic scale where each mark is 10 times greater than the previous one. If we were to try and plot this on a linear scale, this graph would go through the ceiling. So for all of the years that Spartina alterniflora spread, and this is exponential growth, it spread at a constant rate and grew exponentially. In San Francisco Bay, between every 2 points in time, there was an increase in the spread rate, what I have called greater than exponential growth. So we've had an accelerating spread rate, and this is basically unheard of in a pure species whose rate of growth is determined by eons of evolution. They do not change like this. How did this happen? Genes from both parental species and strong natural selection for seed establishment resulted in hybrids that were greater than the parental species in basically everything we looked at. Pollen production viability. This leads to a phenomenon known as pollen swamping, where a single plant preferentially pollinates plants surrounding it. Higher density, higher seed set, including self-fertility. And in addition to the genetic work that told us that it was self-fertile, we also had extensive field studies where we looked at outcross and augmented fertility. So it held true for manipulating the plants and the genetics and the rate of clonal growth. And this resulted in evolution in contemporary time. Within about 20, 30 years, we saw evolution. The way Spartina alterniflora invades is that plants start as seedlings in the mudflat. And this is what San Francisco Bay is supposed to look like, this open mudflat. The plants grow. Eventually, these individual clones, these circular clones, grow together until they get close enough in a meadow to pollinate each other. This is something known as an allele effect, where a plant or an animal can't find a mate. So the whole thing is driven by this self-infertility of alternate flora. What we saw in San Francisco Bay, self-compatible hybrids were able to short circuit the whole process. These individual isolated plants produce the seedlings that colonize the mudflats. Whoops. So to summarize, we have a comparable situations. Alterniflora was introduced into both San Francisco and Willapa estuaries into this open niche habitat, open mudflat, available for colonization. 40 years in San Francisco, 120 in Willapa. In San Francisco, there was hybridization, but there is no native Spartina in Washington, so no hybridization. We saw evolution that was fueled by this genetic variation, resulting in the accelerated spread rates that we measured, whereas in Washington, evolution was constrained by a lack of genetic variation, constant spread rate. So we wondered, what's driving this invasion? Is it the fact that hybrids produce viable, abundant pollen, resulting in pollen swamping? Is it higher seedling survivorship on the open mud, or is it higher clonal growth rate? And the way we evaluated this was using a mathematical model to assess the relative importance of these mechanisms. And the result was eventually the hybrids take over. So in the beginning time, right here at time 0, with all of it being foliosa and a few alterniflora plants introduced, hybridization occurred, and through the generations, both parental species are eliminated. So the pollen production did not play a role in it, but it did increase the frequency of hybrids. What really drove the invasion was the increased hybrid seedling survivorship. Some of the clonal growth contributed, but by far it was increased hybrid seedling survivorship. So seedling recruitment now we know drives the invasion, and it itself is a function of plant factors which are greater than hybrids: flower intensity, seed set, especially selfing seed, local seed dispersal, as we saw in the genetics study, and environmental factors. There have to be safe sites for seedlings to establish, and it turns out a favorable climate. So what is a safe site? What is an unsafe site? Tidal water coming in or flowing out rips seedlings out of the mud. Even without tremendous wave action, the force of the tides moving in and out rips out seedlings. So anything that can moderate tidal forces is going to favor the establishment of seedlings. And here we see a hybrid clone near to the shore. You can see out here another one far from the shore, and these other ones are far from the shore. But in the lee of this plant, there was tremendous seedling establishment. We did not genetically analyze this relationship, but I'm willing to bet that this is the mother of many of these seedlings that established in her shelter. Another safe site are restoration sites. These are former salt ponds. They add sediment to it. They deliver the tide through channels, not directly, and they want to turn it into a tidal salt marsh. So here is Cogswell Marsh and Hayward in 96. These red circles, these red blotches, those are plants, and this gray area is open mud. We found that tidal channels were lined with hybrid cordgrass, and so when they shed their seed, they delivered it right to these salt ponds. In fact, we had a poster, if you build it, they will come. And they did. 1996, then there was an El Nino year. And then in 98, we saw this tremendous spread, recruitment, lateral spread of clones in Cogswell Marsh. So something was going on with the climate. The El Nino southern oscillation, El Nino, has rainfall twice average. And we found that whatever the genetics of the plant, high rainfall promotes clonal growth, flowering, and seed set among all plants. They like the fresh water. We, Christina Sloop and I, we walked mud flats looking for seedlings. And in 2003, we found seedlings. And this is seedlings per mud flat. Roberts Landing, Hayward, and Alameda Island. In 2004, we saw this huge increase in seedlings, and then a complete absence of seedlings in 2005, which was a La Nina year. So clearly climate contributes to the safe site. And we concluded that seedling recruitment is episodic due to climate and is site dependent. So the invasion dynamics, seed production is key. Seedling establishment depends on this dispersal to safe sites. And this informs us on how to control these hybrids. The elimination of all hybrids to reduce hybrid seed set and pollen swamping. Controlling hybrid seed production and seedling establishment by reduction safe sites, eliminating those safe sites, and vigilance, especially during the El Nino years. There is a huge restoration project in the South Bay called the South Bay Salt Pond Restoration that we'll probably be hearing more about in the future, how they were swamped by hybrids because the hybrids were not eliminated, as you will hear in the video that follows this. Hybrids were not eliminated. Of course, there are more than just hybrids growing over open mudflat. Who cares? There are a lot of ecological consequences to this invasion that are detailed in this proceedings of 2004. And I would like to thank all of the postdocs, grad students, undergrads, and technicians, all of us mentored by Don Strong to do this work. So I've spoken too long. Are there any questions? You can unmute.
This is, in one of your slides, it looked like the colony was out in the middle of the bay.
Yeah, well,
was that right?
Out on a mud, that was correct. It's out on a mud flat about 200 meters from the shoreline, 600 feet.
So, but these plants don't, they like a mixture of salt and fresh, but they don't grow in salt water, do they?
They do. They grow in seawater.
Okay.
They grow, yeah. Like, yeah,
Is it
they grow in
possible that a whole island of them could break loose and float away down the bay
No,
and out?
They are well into the mud, and they continue to accrete sediment as it washes in from the tidal creeks and whatnot of the bay, and gradually the elevation of the marsh increases and increases. The changes are not irreparable, but they do take a long time to, that elevation takes a long time to go away once the plants are killed.
So
Anybody
Deb,
wonder how? Yeah.
So Deb, I was wondering about the hybrids because you have introgression. So I'm wondering if you had seedling recruitment in varying elevations.
Yes.
So, okay. Yeah. So that would also increase
Wherever
the spread.
wherever the seedlings can escape that tidal action down, but you saw islands of dual cordgrass plants that were able to gain a foothold, root hold, out in some fairly deep mud, really out there quite a ways on the mud flat, and then they act as recruitment of further seeds that they themselves produce, as I also showed you.
So they're mostly the seed dispersal. Is that mostly just by floating around, or do they do some
But-
wind action.
They float on the tide, individual seeds float on the tide, and it's also been shown in Washington, those culms, those grass stems break off at the end of the year,
Ah,
some of them containing ripe seed, and they form rafts, and the whole raft grass stems float long distances.
God.
Yeah, so there's this potential for long distance dispersal, not just local.
Wow. Wow.
Okay, any more questions? And if you have any comments, email me and say, damn, you talk fast, or whatever it is you want to say. Okay, so I'm gonna try and put this on. Help me. Let's see, how do I'm gonna put a video on. So I need to share my screen.
Yeah, and
You
then,
took your slides off, so that's good.
Yeah.
So
That's right.
show us.
then click on your movie.
Okay,
Yeah,
can we
show-
see that?
It's coming up.
It's got,
Give
there
us a
it
second.
is.
And now you want to click it again like you did before, make it your whole screen.
Okay, I did.
That's good.
Now,
And
this was presented to a weed group last year, and I've cut out all the introductory blah, blah in the interest of saving time. Okay, so we're going to jump right in.
From some of the work that one of the partners, Drew Kerr, has done from flying his drone around some really interesting pictures of marshes from the air, showing, for instance, how it looked, several years back, and then how it's looked after revegetation with native Spartina. And the ability of Spartina to spread is pretty amazing over several seasons. How's it looking now, Jeannie?
I got it, yay!
Right, take it away.
So first, before I get into the invasive Spartina species, I wanted to talk a little bit about why native tidal salt marshes are important. So when you're flying into San Francisco or Oakland airports, or when you're whizzing across the San Mateo Bridge or the Dumbarton Bridge in San Francisco Bay, you may have noticed, I'm sure you have noticed, the sinuous blue and green of the salt marshes and maybe some of the reds of the former salt ponds. Or maybe just you smelled that funny sulfury smell on a summer day when you lowered the window down. All of those are sort of salt marshy kinds of things, and salt marshes are super important to the health of San Francisco Bay, and also to all of us who live on the edges of the bay. We live and work and play on the bay. So why are salt marshes important? They're highly productive ecosystems that are really rich in biodiversity. They're also important carbon sinks, so they're helpful in combating climate change. They can also help protect coastal areas from natural disasters, acting as a buffer against storm surges and erosion. Essentially, they're the upper vegetated portion of mudflats. So if you think about it, when you're out there looking around the bay and the tide goes out and you see all the mudflat, the upper portion of that is sort of where the tidal marshes are. They're sandwiched between the marine and the terrestrial ecosystems. And sometimes they provide the connection between those saline waters and the fresh waters. So they're super important critical habitat for a bunch of different aquatic and terrestrial organisms that feed and find shelter and breed in salt marshes. Salt marshes also act as high tide refuge for birds that are feeding on the adjacent mudflats. And they also provide really valuable ecosystem services of significant commercial and recreational value to all of us. And one example of that is they really help protect water quality by filtering contaminants from our urban runoff. And globally, we've lost 25 to 50% of our salt marshes. But this graphic kind of shows you in our San Francisco Bay Estuary, we've actually lost more like 85% of our tidal wetlands to human development. And since, so the left-hand side, this is a really great graphic that San Francisco Estuary Institute put together. On the left side, it shows extent of what we had in tidal marshes, estimated about 200,000 acres. And then today on the right side, you can see the really light green is where we have existing tidal marshes, or prior to us starting to do marsh restoration. And the nice thing about this graphic on the right is it really shows the effort that the conservation community around San Francisco Bay has in already starting restoration projects, and then the dark green are some of the ones that are proposed in the future. And the Baywide goal is to get us up by 100,000 acres once all the restoration that's planned here is finished. So thinking about the fact that we've lost 85%, the San Francisco Bay Estuary is still the most expansive area of salt marshes on the West Coast. And it's the second largest estuary in the entire US. So it's still super important. The bay's tidal marshes are extremely productive habitats, and they create a lot of nutrients and a lot of organic material that supports the bay's food web. Marsh plants also they provide protected areas for many animals for foraging and breeding and nesting, including a number of endangered species. It's also really important to have wintering habitat for waterfowl and shorebirds. And of course, we all know San Francisco Bay is a really important stopover area for an estimated more than 1 million birds each year that migrate along the Pacific Flyway. And this slide just shows you, thinking about all that habitat loss, of course, the critters who lived in those habitats have also decreased in number. And these are just a couple of them that we deal with in our project, salt marsh harvest mouse. And these three are all protected by federal and state Endangered Species Act. Salt marsh harvest mouse, California ridge-raised rail, and California black rail. So what are some of the continuing threats to our salt marshes here? And like all salt marshes around the world, the biggest threat is climate change. And, our coastal communities are threatened by sea level rise, and that also applies to our salt marshes who are at the edges of our coastal communities. With that increasing water level, the vegetation is going to get inundated more frequently, and it may or may not be able to persist. It's the effect of drowning in salt marshes. In addition, there's this shifting precipitation patterns that we're seeing as part of climate change, with less winter snowpack, possibly more winter rainfall. Currently,
Deborah, we lost volume. We lost sound.
marshes are also known to sequester carbon, so helping with
Is it better now?
greenhouse gas emissions. And then something that we've all discovered
Yes.
in this past year is that open
I muted
recreation
myself.
is critical to our
Was not
mental
the thing.
health. And around San Francisco Bay, we have the San Francisco Bay Trail, which opens up these areas to us and allows us to get out and really enjoy that open space. So this slide is really just sort of a little tiny window into bay conservation and sort of the little pieces that are relevant to the establishment of the invasive Spartina project. I'm sure a number of you are from Northern California, so you may are probably familiar or have heard of the Reaver plan. With the 85% of the tidal wetlands that we lost, we were going to lose more if this plan had gone into place. Mr. Reaver first proposed it in the 40s, and it basically was 2 giant dams that would have gone across roughly where the Richmond-San Rafael Bridge is today, so sort of in the northern bay, and then where the Bay Bridge is, which goes over to San Francisco from the East Bay. And behind those giant dams would have been 2 very large freshwater lakes. And in addition, it had called for 20,000 acres of additional fill with a shipping channel and locks leading out to the bay and then the ocean. And luckily, it was completely infeasible, and the Army Corps engineer declared it so in the 60s, but it was around from the 40s to the 60s. And so as part of that, 3 women who lived in the East Bay saw this plan and looked out their windows and thought, oh, gee, my home here in Brooklyn, they're going to fill all in front of me. And it basically catalyzed the Bay conservation movement. And the women started Save the Bay. And as part of that, they in turn then helped get past the first wetland protection legislation in San Francisco Bay, which included the establishment of the Bay Conservation Development Commission, which is the first coastal zone management agency that was really charged with protecting tidal wetlands and specifically stopping fill of wetlands. And so the kind of vision that the state had of changing our bay from being sort of a dumping ground into what we think of it today as this treasured resource, open space, and home for critters and everything. It's really started here. And as we started to think about, you know, we've lost all these wetlands and want to get them back, the first wetland restoration projects were implemented in the 70s and the 80s. And as part of the overall conservation movement and the sort of importance of coastal resources coming to mind, the Coastal Conservancy was established in the mid-70s, and to support projects that protect coastal resources. And while this is all happening in the 1980s, the East Coast species of cordgrass called Spartina alterniflora was introduced in a restoration project by the Army Corps. And kind of, I won't be talking about Spartina alterniflora, I'm going to talk about hybrid Spartina, and I'll tell you how that happened in a minute. But the hybrid Spartina spread from that initial site, and so landowners around the bay started trying to take care of it and realized pretty quickly that you know, a plant that can move around with the tides and the tidal system, it's one landowner can't deal with it. You need to have a really collaborative, coordinated regional effort, and that's where the invasive Spartina project came from. Okay, so who are we and how do we do the work? Like I said, the State Coastal Conservancy is involved as the co-lead on our project with the U.S. Fish and Wildlife Service, and Calypsy and Olison Environmental manage and do the work on the ground. We also have, you know, 200 partners around the bay. You can imagine anywhere that the bay touches lands around the bay is a potential partner for us, because that could potentially be habitat for invasive Spartina. Those partners include, you know, everything from private landowners to cities and counties and mosquito abatement districts and water districts and local, state, and federal agencies. And it's pretty much anybody who has jurisdiction or owns land near the edge of the bay. So first, before I talk about the invasive Spartina, I really wanted to touch on our native Pacific cordgrass, Spartina foliosa. Also, due to a fairly recent taxonomic revision, I think everyone's sticking with Spartina in the near term anyway, but it's just a fun name to say, so I thought I'd say it. A perennial grass in the grass family Poaceae, and it's a rhizonomous grass. So what that means is that it can spread both by seed, but it also has rhizomes, you know, underground root structures that grow laterally and tillers that will come up so that it can spread clonally across a mudflat as well. Distribution of the native cordgrass, it's limited to California and Baja California, and it's really a foundational species in our low marsh here in Cisco Bay. It's an early colonizer. It can come in via, you know, pieces of the rhizome can actually move around the bay and plant themselves, and a new plant can come up from the rhizome. So it doesn't just spread from seed, it can also spread from rhizomes fragments. It also grows in a really narrow elevational range in tidal marshes, and you know, it kind of stays in its lane essentially. And I included this little diagram. It's a marsh zonation diagram in San Francisco Bay in the right-hand corner of the slide. And you can see, you know, go lower elevations, you're down in kind of bay waters and the subtidal, you come up into the mudflat, and then that one up is the low marsh where vegetation can start to come in. And that's where Spartina foliosa, the native cordgrass. Above that, you get into what we think of as the tidal marsh plain, where perennial pickweed grows and other plants, and you have your salt pans and your channels. And then higher elevation, you get into the ecotone, which is sort of that transitional habitat between the tidal habitats and the upland habitats. And the one thing I wanted to say about Spartina foliosa in particular is that I wanted to point out that in San Francisco Bay Estuary, over 99% of the cordgrass that we have is the native cordgrass. All right, so what are the invasive Spartina species that we have in San Francisco Bay? I'm going to talk first a little bit about Spartina densiflora, which is up in the right-hand corner. It's a perennial bunch grass that was originally introduced into the US, into California in Humboldt Bay via the timber trade in ship ballast. And then it was introduced locally in San Francisco Bay as part of a restoration project in Corte Madera Creek watershed, which is in Marin County. It was mistakenly thought to be a form of the native cordgrass. And then in the left-hand side, we have Spartina anglica, which is also the result of that same restoration project. And it's limited to the original introduction site. There's very little of it left. And Spartina anglica, also known as English cordgrass, is actually a hybrid between the East Coast native for US, Spartina alterniflora, and an English species called Spartina marittima, hence the common name English cordgrass. And then in the lower right-hand corner, we have Spartina patens, which is also called salt meadow cordgrass, which is only found at one site in the Benicia State Recreation Area. And these three species I've just talked about are very, very close to eradication. There's extremely small amounts of them left in San Francisco Bay. And our big problem that we still have is the one in the middle, which is our hybrid Spartina. And hybrid Spartina is a hybrid between the East Coast native Spartina alterniflora and our West Coast native Spartina foliosa. I'm going to talk a little bit more about that hybridization now. So how did it happen? Like I said, the East Coast species Spartina alterniflora was introduced here in the 1970s as part of another restoration project by the Army Corps of Engineers. They were looking at ways to stabilize dredge spoils, a bank stabilization project. You know, we know a lot more now about what the possible unintended consequences of introducing a novel species into a different ecosystem. So the hybridization of the native cordgrass with the East Coast cordgrass probably happened sometime in the 80s, and it kind of lurked for a little while and no one knew what was going on. It was eventually discovered in the 90s. And in the meantime, that original hybridization happened, and then there's been a bunch of additional events where there's a lot of back crossing that happened, and which produced a lot of different forms which have some transgressive traits, meaning that they can exploit a lot more of the tidal marsh niches than the original native species. Okay, so this is a little more on the hybrid. On the left-hand side, the illustration is Spartina foliosa, and the other illustration is hybrid Spartina. And then the photo that shows the two different plants, you can see Spartina foliosa on the left and hybrid Spartina on the right. And so something that happens with hybrids quite frequently is hybrid vigor, which means that the hybrid has more robust characters than either parent species. And in this case, the hybrid Spartina tends to have, it tends to be taller. It tends to grow more densely and have greater cover, which means that it ends up outcompeting pretty much all other tidal marsh plant species. It also can grow, the inflorescence is longer, clearly you can see in the photo, which means that it can produce a lot more seed. It also, the biggest problem for us really is that it grows in a much wider tidal range than the native foliosa. So I included that marsh zonation diagram here again, and you can see on the left side, Spartina foliosa are native in its really narrow low marsh zone. And on the right side, you can see the hybrid Spartina, which can grow much lower in the tidal frame down into the mud flats, also up across the marsh plain and even up into the ecotone. So it really, hybrid Spartina basically threatens all of those tidal marsh habitat types. So I just talked a little bit about why hybrid Spartina is a problem. You know, it can degrade the existing native tidal marsh ecosystem because it comes in and outcompetes all of the other tidal marsh species. It also changes the hydrology because it's you can see this photo in the top here. It gives an example of this where it's just really tall, dense plants that end up accreting more sediment, and they can grow lower into these channels, and eventually you can see that this channel would just be choked with vegetation, and you end up with a monoculture of hybrid Spartina across the site. Also, because it can grow lower in the tidal frame, it marched out across mudflats, which basically excludes foraging habitat for shorebirds. And there are changes in the invertebrate community as well as a result of that. UC Davis did a bunch of research on the invertebrate community and invaded these hybrid Spartina meadows versus unvegetated mudflat, and found that, you know, the number of invertebrates available in the surface muds decreased by over 7%. And the types of invertebrates that are available also changed. So you can envision shorebirds foraging in the mudflat and how far their bills can go in. They really, you need those surface feeders. And it turns out that when you have a hybrid Spartina meadow, the surface feeders kind of go away and you get these more subsurface feeders. So it also was impacting their food resources. Hybrid Spartina also endangers native Pacific cordgrass. Pollen swamping, much larger inflorescences. If you've got hybrid growing next to the native cordgrass and the pollen goes out there, it can get onto the native cordgrass and end up making the native cordgrass produce hybrid seed, as well as the hybrid outcompetes the native. So eventually, if left unchecked, hybrid Spartina would likely cause the extinction of Pacific cordgrass. I talked a little bit about this in terms of the hydrology, but it also can reduce flood control capacity. As you can imagine in this picture up here, when it grows across the channel, and it's a very dense growing vegetation, that is going to impede flood flows. And so that is possibly going to cause higher risk of flooding upstream. Mosquito abatement districts around San Francisco Bay are actually some of our major partners, because they have found that hybrid Spartina meadows slow down water, create ponding. And when you have ponding, you have more mosquitoes. We have more mosquitoes, you have high risk of diseases like West Nile virus. Also, for all the reasons I listed above, hybrid Spartina also causes failed native tidal marsh restorations, because if hybrid Spartina is present, it's going to be one of the first plants to come in and it's going to outcompete everything else, and you're going to end up with a hybrid Spartina meadow, when what we want from our native tidal marshes is all of that complexity that we see with the channel, the sinuous tidal marsh channels, the mudflats, the ecotone, the marsh plain, the salt pans, that basically can just become a monoculture. Okay, so I threw this slide in here to show how much hybrid Spartina is an ecosystem engineer. So this is showing a site where you can see in that red rectangle, just a few hybrid Spartina clones right off the edge of this creek mouth. And in less than 20 years, the amount of sediment accretion and more hybrid Spartina growing, and sediment accretion, more hybrid Spartina going, and you can see that Spartina meadow has moved out significantly into the mudflat. And you can sort of envision what would happen if this had been left unchecked. If we hadn't started controlling for it, it probably would have covered a major portion of our mudflats in San Francisco Bay. So in response to the threats of the estuary, the invasive Spartina project was initiated to coordinate estuary-wide Spartina control efforts. And we began full-scale implementation treatment in 2005. And we currently do annual surveys in potential habitat for hybrid Spartina of over 70,000 acres that informs our treatment program. And this slide just goes into a little bit more detail about the extent. You can see where hybrid Spartina could be distributed around the bay. And from 2005 to 2020, it shows that sort of how things have changed over time. In 2005, well, I should describe the colors. So the dark green indicates places where there's no invasive Spartina, and the dark red indicates places where originally there was more than an acre of hybrid Spartina. And as you can see over time, those red sites go away completely, or not completely, but we're down to very few of them, and we've basically reduced the amount of hybrid Spartina around the bay significantly. And to give that in a more graphical representation, here, we started with roughly 805 net acres in 2005. We're down to 33 acres in 2020, and that's a reduction of 96% estuary wide. And so we're kind of that critical. We need to keep continuing our project so that we can complete eradication, so to support all of these regional native tidal marsh restoration projects that are coming online. And with that, I'm going to turn it over to Lindsay, who's going to talk about how in the world do we do this?
Awesome, thank you, Jeannie. Hi everyone, I'm Lindsay Demikis. I'm one of the biologists that gets boots on the ground for ISP at Olson Environmental. And I am going to talk today about our different programs. So we'll start out talking about our inventory program, where we go out and actually ID and map invasive Spartina around the bay. Then we have our treatment program. So we go out and we treat and remove the invasive Spartina that we've found and mapped. We have our ridgeways rail monitoring program, where we monitor the population of ridgeways rails throughout the estuary. And we have our revegetation program, which is spearheaded by Jeannie. We add plants into areas that we've removed hybrid and bolster marsh habitat for rails and other species. So I chose this picture. It's a great photo taken from a helicopter by Drew Kerr over Bear Island, which is one of our biggest marshes in San Francisco Bay. And you can just see the vastness of just this small part of Bear Island. And there are two biologists down there. Looks like they're probably doing some inventory. So walking sort of these tight track lines up and down these marshes to look for hybrid Spartina and get it mapped so that we can go out and treat it. So with that, next slide, Jeannie. So OEI biologists inventory up to 70,000 acres of the San Francisco Bay year to year. We don't get to every single one of those acres every year because we have sites that don't need to be looked at as often, but there are years where we get pretty close. So we walk around the bay with handheld GPS units that help us take accurate locations of hybrid Spartina patches. And the benefits of this extensive inventory is that, first of all, it saves time because the treatment crew only has to come out and visit features that have already been mapped. It reduces impacts to sensitive habitat. So biologists are able to lead treatment crews to mapped features and minimize walking through marsh and disturbing sensitive habitat more than is necessary. It also gives us the time to thoroughly inventory the marsh and identify the hybrid Spartina, and that allows us to save as much of the native foliosa as possible. Just to throw it out there, this is not an easy task because Jeannie didn't really go into hybrid Spartina features as in they vary. So usually hybrid Spartina is bigger and denser and more robust, but that's not always true, especially based on where it lives in the bay, also where it is in the marsh itself. And there are just lots of different factors that determine what hybrid Spartina looks like. Time of year matters. So it's definitely an art form and one that we are all perfecting on a year-to-year basis. And we do an extensive amount of genetic testing every year. So we take approximately 500 genetic samples each year. And we tend to do this in areas where we really want to save the foliosa. So if we have patches of hybrid Spartina that we're not quite sure are hybrid, then we'll do genetic testing, and that helps us inform treatment for the current year. And it also just helps us inform inventory for future years. Next slide, Jeannie. So this is a graphic you've already seen from Jeannie, but I thought I'd spend a little bit more time talking about it and emphasizing the extent of this monitoring that we do widespread throughout the San Francisco estuary. So we have 220 sub areas that represent these 70,000 acres of habitat. And in 2020, OEI mapped 33 net acres of invasive Spartina. That's down from over 800 acres of invasive Spartina 15 years ago. So for context, that 33 acres is 0.0005% of those 70,000 acres. So that's a pretty small number. Things are definitely improving and we're doing a great job, I think. And then Jeannie already broke down what the different colors mean. But basically those greens and yellows are good and the oranges and reds are not as good, but definitely seeing a lot more as we're moving forward. And Jeannie, if you'll give the slide a click, very good. So the little channel adjacent to this star that looks like a little finger kind of coming out and pointing at the star is AFCC, that's the Alameda Flood Control Channel. And that's our original introduction site of Spartina alterniflora, or the beginning of hybrid Spartina that Jeannie discussed earlier. And you'll notice that that channel is green, which means that there's not a lot of hybrid Spartina there anymore. Done a great job of eradicating her. I think there's still a little bit, but we're looking really good there. So yeah, next slide. So then just to dive a little bit deeper into those numbers, as of 2020, 151 of our 220 ISP sites that had historical infestations of hybrid Spartina now contain under 10 meters squared. And the total in that area is 186 meters squared net cover of invasive Spartina. So those are the numbers as they stand now. And just to give you an idea of, I know probably most people here know what a meter squared looks like, but when we go out in the marsh, we identify plants and then we map them, and we have to give a really good estimate of how much hybrid Spartina we just mapped, so that we know how much chemical or crew or whatever to bring out so we can do our treatment. So shoulder to my fingertips. So if you just go like idea of what a meter squared looks like, and then you can kind of have an idea of what 186 meters squared looks like. So these sites that we work on encompass tens of thousands of acres of marsh and mudflat. So it's a pretty impressive amount of space. Next slide. Okay, so we're going to talk a little bit about how we go out and treat invasive Spartina. So we have a variety of tools in our toolbox, and the reason that we do is because depending on what plant we're treating and what's going on in our treatment program and how we're doing, we can adjust our treatment as needed. So for example, Spartina densiflora, as mentioned earlier in the talk, is a bunch grass, which makes and then we also use tarp sometimes. So like digging and tarps are the two ways that we can, like tarping, are the two ways that we can eliminate invasive Spartina without using any chemicals or herbicides. And those are pretty effective for certain types of Spartina. And then for our hybrid Spartina, we use a herbicide called imazapyr. And that is because, as Jeannie was discussing earlier, hybrid Spartina is rhizonomous. So if we dug it, we have the potential for missing pieces of the rhizome, and then the plant will come back the next year. So our hybrid Spartina treatment program starts getting started right about now, and year to year we treat about 150 sites throughout San Francisco Bay. Next slide. Next slide, Jeannie.
Uh-oh.
Hold on, I'm trying. It's frozen. Figures, right? Let me. I guess just hitting the button won't really make it do anything. I think I might need to stop sharing to get it to go forward.
Have you tried using the down arrows instead of return?
That's what I'm using.
Okay.
Down arrow, side arrow, any arrow.
If you're on two screens, make sure you click on this screen and then try the down arrow.
Yeah, I haven't moved off of the screen, so.
Weird. Okay.
I know, right? Let me see. Wow. I can't even get out. My screen is frozen. I'm so sorry, Lindsay.
That's okay, no worries, technology.
I'll use this moment to encourage folks to think about any questions you may have and enter them in the Q&A, and we'll bring those up at the end of the talk.
Jeannie, I also have the PowerPoint, so I have it ready. I can open it if that's helpful.
Yeah, let me see if this works. I'm just going to reopen it like that. I'm going to do this. There, let me see. Oh,
Yay. Awesome. Okay, I'm gonna power on. We're looking at eradication of densiflora. So that's the Spartina species that we generally dig instead of using herbicide. Densi sites are historically up in the Marin part of the bay. So we go there twice annually. The first time we go in June when the plant is flowering, and that makes it pretty easy to find. And the second time is in January when the native pickleweed hits the nest. So it's brown, so it contrasts really well with the Densi seedlings that are more green that time of year. It's really great to find them in January because once they go to seed, we have a little bit more of a problem because the seed bank for Densi is 5 years long. So if we miss a plant like this one that's already gone to seed, there's a high likelihood that we're going to find a plant there in the next few years. So we try to catch them as many as we can in that January time frame. So we found 66 seedlings in 2020, and we've reduced densiflora up in that part of the bay, and the bay in general, because that's really only where we find it, by 99.9%. So really great number. Next slide. So imazapyr is the herbicide that we use to remove hybrid Spartina around the majority of the bay. The EPA considers imazapyr practically non-toxic, and concentrations in water after treatment are far lower than any amount that would be of concern for wildlife. And one of the reasons for that is because imazapyr targets plant amino acids specifically. So imazapyr kills plants by preventing the synthesis of certain necessary amino acids that are produced by plants, but not by animals, which is what makes it pretty safe for surrounding wildlife. And this treatment takes place with close monitoring by biologists. So you can see in the photo that I think that's probably my guy Richard out there spraying, and he's got a biologist keeping a close eye on him and has cleared the area beforehand to make sure that there aren't any animals there. And if we do encounter a rail or a salt marsh harvest mouse, we leave the area and get back to that patch another time. So next slide. So the invasive Spartina project is a pretty textbook example of a conservation dilemma in a way, because we have endangered species versus invasive species. So human development has led to this massive loss of tidal wetlands in SF Bay, and wildlife that's dependent on those wetlands is now endangered. So human action has then led to the introduction of highly invasive species, which ironically do help create habitat for these creatures that we care about. So how do we balance removing invasive species while protecting endangered species? Well, the answer for us at ISP is adaptive management. So treatment, restoration, surveys to assess population, and then reconsultation for future treatment. Next slide. So here's our California Ridgeways rail, and the photo on the left shows a great example of some hybrid clones that are creeping out and making the marsh bigger. They've created brand new marsh where there was none before, which of course our rails like having habitat, lots of habitat. And then also the hybrid grows tall enough that it provides great cover at high tide. So it's honestly like a great plant for these birds, but it's outcompeted our native Spartina. So what happens is we go out, we're taking out the invasive Spartina, and because it's outcompeted our native is not there to take its place when it's been removed. So removing invasive Spartina creates this deficit in ecosystem function. So that function that's been temporarily provided by hybrid is usually provided by native plants, but they've been outcompeted. Next slide. So how do we keep track of the rail population so we know if our restoration efforts are impacting the species? We conduct call count surveys for Ridgeways rail between the months of January and April at the start of the breeding season. And Ridgeways rails are a secretive marsh birds. So while we see them fairly often because we're out in their habitat, a member of the public is not likely to see one probably ever, unless you're very patient and have some really good binoculars. But if you're out walking in the marshes in like right around dawn or right around dusk and you hear this sound, then that's a rail. It's calling not to you, but to one of its fellow rail members trying to communicate something, probably that it wants to mate. But yeah, definitely if you're interested, it's a really distinct call, and it's cool to listen for around the marshes of the bay. Next slide. So despite the fact that we are removing habitat, we are seeing rail call counts increase over the last decade. The data here was collected by ISP and our partner organizations that include USFWS and Point Blue. And this increase is due to several factors, which of course includes just bay-wide restoration of tidal marsh wetlands. So we're seeing wetlands where we didn't have them before. We're breaching levees, we're creating new habitat for them, not just OEI, but lots of different organizations and are working on that. And then also just this successful revegetation of places where we've removed hybrid Spartina, and we're also adding habitat to places we haven't removed hybrid Spartina, finding great places to add some refuge plants for rails and other species. Next. So the star of the show, this Ridgeways rail, is about the size of a chicken, And there he is at the top, and he's really the center of our restoration program. So one of our main challenges is balancing hybrid Spartina with the need to protect habitat for these rails. And ISP's restoration program has grown out of that challenge. So that's why we've started our re-veg program. And the main goal of the program is just to rapidly enhance tidal marsh habitat to benefit ridgeways, rails, and other tidal marsh dependent wildlife. Next slide. So ISP's re-veg program was started in 2011, and we just completed our 10th year of habitat enhancements. This program is guided by informed by our tech advisory committee and implemented at a Baywise scale. The program focuses on critical components of rail habitat, and we actively use adaptive management to learn from our enhancement efforts. A great example of this is we've learned that caging patches of new Spartina foliosa seedlings helps protect them from grazing from Canadian geese. So, and we learn where that's important and where that's not based on how our re-veg program did in the beginning, and learning where we need to cage and where we don't. Next slide. So this map shows the 40+ sites where we've implemented habitat enhancement throughout SFA. The blue areas of the map show planting areas, which is primarily Spartina foliosa and marsh gum plant, Grandelia stricta. And then orange shows where we've added high tide refuge islands. The photo down there at the bottom shows ISP staff and probably some of our contractors out there doing some marsh gum in the ground. Next slide. So here are some great examples throughout the Bay of places where we've done some successful revegetation. So the left is, let's see, the top left of this slide here, the two that say 2013 and 2017 are those photos. Thank you, Jeannie. Those photos were taken in Eden Landing, and it was an early restoration project where it was mud flat before, and then we successfully planted Spartina and have this great foliage band going along that channel right there, which is fabulous habitat. And then the photo below that, still on the left-hand side on the bottom, that's AFCC. So that's our original alternative flora and then subsequently hybrid introduction site that we've eradicated most of that hybrid Spartina and planted this great band of foliage that's grown and become just this awesome stretch of habitat along that big major channel. And then on the right-hand side, you'll see two examples. Both of those were taken along the Hayward shoreline of where we planted marsh gum plant along a couple of channels and just watched it absolutely explode over a couple of years into really phenomenal habitat. Next slide. So just one last example of some great success we've had with revegetation. So this slide shows a photo that was taken in 2011 after this unvegetated mudflat was opened back up. And this site is pretty far from any local source of native cordgrass, which is the plant that we would expect to colonize given the elevation of this mudflat. So we wanted to jumpstart this trajectory of this site to open mudflat from open mudflat to potential habitat for ridgeways rails. So we did active planting here, which you can see in the next slide. So here are some photo points that have been taken. So we go back to the same location year to year and take the same photo over and over again. And so you can see we have 3 photo points here where there's basically bare mud beginning, and then a few years later, really happy, healthy looking Spartina foliosa. And then on the right-hand side here, we've got a kind of a bird's eye view. This is actually what like our GPS units look like in the field. So those green patches are plantings, probably from multiple years. And then we also have the locations of ridgeways rails that we've heard during call count surveys. And then that A, B, and C corresponds to those photo points on the left side. So that's where those photos were taken. So we go back to those areas from year to year and see how they're doing. Next slide. And now here's just an overall shot of what that marsh looks like. So you saw 2 slides ago that it was a big open mud flat, and now it's a beautiful, happy marsh. And the best thing is that the population at the site is rapidly expanding. We detected 0 birds in 2018, 2020, and 13 in 2021. 3 years we've had 13 ridgeways rails move in there. Next slide. So sort of to wrap up here, the invasive Spartina project is a constantly moving target for us and for all of our partners. And we continue to remove invasive Spartina, restore native marsh habitat, and monitor the California ridge-raised rail. And we hope that working toward this balance can help create and protect healthy native marshes that are more resilient in the face of sea level rise and climate change. Next slide. With that, we'd like to thank our partners. This is just a small spattering of them. We have so many, but we definitely couldn't do any of this great work without all of our partners and collaborators. So thanks so much. That's it.
Wow, yes, sorry, a little slow on the unmute trigger there. That is amazing. Having tried to walk in a mud flat once just to sample some Spartina back in state parks days, I know what a challenging environment that is to work in. So Nikki, what sorts of questions do we have from folks?
Yeah, we have a few. Sarah wants to know, is there anyone monitoring invasive Spartina in the legal delta? If so, do you know where it needs to be managed?
Yes, we're working, recently added the Sassoon Marsh area as a fairly new monitoring region for us, based on an observation that someone had. Like stepping back a little bit, thinking about freshwater and saline waters, in the farther you go upstream, Spartina species end up dropping out because you get into freshwater species and they tend to compete better. And so you don't really find native cordgrass up the San Francisco or the Sacramento River Delta very far. And so we've looked over the years, but not that much. And recently, I think with the increasing drought years, it's probably starting to move up a little more. And so we've started to detect a few patches. There's not very much of it. It's mostly under control. And I'm trying to remember where. So there's a few patches in the Motco area, if you know where Motco is, essentially. I think that's about as far upstream as it goes right now. But we're actively treating them and they're almost down to zero, I believe, right, Lindsay?
Yeah.
It's a huge area to suddenly have to inventory. We're trying, you know, for the last 3 years, I think we've tried different methods, you know, whalers, it's always windy up there in the Delta, trying to get out to these areas, you know, getting permission to access duck clubs. It's been an interesting undertaking to start getting your handle on Sassoon Marsh area.
All right, great. We had a couple questions about Amazipar from Francis and Naresh. Saying Amazipar is on the California groundwater protection list. Even though the applications are on saltwater marshes, are you folks monitoring its presence in thresholds and adjoining groundwater tables? And along with that, Francis wants to know, what is the half time for Amazipar? Does it, I think, harm any environmental species?
Good questions. I am not the treatment program manager, so I am almost afraid to answer these questions, but okay, say again what the
If I understood correctly, it's Amazipur is an herbicide and it's being applied to the marsh. I know a little bit from hearing from Drew, the treatment manager, that is applied at a certain tide level and that the half-life in water is quick.
right.
So
Yes.
you want to apply when you have exposure and it's not going to get washed off right away. And then they also do water quality measurements for the NPDES permit all the time and have found basically none. So that's as far as my knowledge goes, but I don't think of it.
Okay, so I hope I'm rejoining the main group. Can you hear me? Hello?
Yes. Yes.
So
we
I
hear
thought,
you.
oh, good. So I thought I would pause it here. We can certainly listen to more questions. I can address questions you might have had about Spartina biology, or we can say goodnight. So what do we feel like doing? Do you want to hear some more questions? There's about 10 minutes more on this video. I can't hear you. Shall I play it some more, or are people here? Hello? Okay, well, I'll shut it
We can-
down then.
I think we can skip the rest of the questions.
Okay, let's stop that share. All right. So I want to make some just a couple comments. The first 10 years of the invasive Spartina project cost $20 million. I don't know how much it's cost in the last 12 years.
Oh, wow.
So I know that we tend to think of the weeds in our garden. That's a weed problem. We look at yellow star thistle or stinkwort. This is a weed problem that spread widely across the bay, creating multitudes of problems. A weed problem that we don't normally consider this kind of a vast weed control problem. So I wanted to present that to you guys today. I know we don't live around the bay, but we get there often enough. And you can appreciate the complexities involved in controlling a weed that is an environmental engineer, as Spartina hybrids are, and how the eradication has then been followed by restoration for an endangered bird. So it's a complex, expensive process. So are there any questions for me? Right. Well, if you have any comments, please feel free to email me. And thank you, Lisa. And of course, it's available online if you wanted to watch it, watch the video again on how to deal with the Spartina. So thank you all for coming.
Thank you.
And
Thank you, Debra.
September. Okay, thank you. We will be talking about the garden that is at the south side of the Placerville Library. The 3 people who were instrumental in designing, implementing, and actually planting that garden will be talking about that process for our September meeting. So good night, everyone.
Night, Deb.
Good night.
Good night.